So FUS protein does not need to be introduced, so very briefly, mutations in FUS account for about 5% of familial cases and very rarely found in sporadic cases of ALS, and FUS positive inclusions are found in the affected nervous tissue of patients with ALS and PTLD and some other more rare conditions. Here. And the protein briefly comprises a prion-like domain at its N-terminus, and RNA-binding domains are clustered in the C-terminal part of the protein. And the hotspot of mutations at the very C-terminus nuclear localization, which you see here. And now these diseases are now combined under the name of FUS proteinopathies. So mutations in FUS affecting its nuclear localization signal cause cytoplasmic redistribution of the protein, at least in cultured cells, so which can cause either loss of nuclear function or gain of toxic function in the cytoplasm. An important step forward in our understanding of disease pathomechanisms in FUSopathies was the discovery that cytoplasmically mislocalized FUS can be recruited into stress granules, which represent cytoplasmic foci composed of RNA and protein, and which serve for protection of cellular RNA under severe stress. However, in addition to recruitment into stress-induced stress granules, FUS is often found in cytoplasmic aggregates. These are just a couple of examples here from different studies, and these are often referred as pathological stress granules. However, the exact relationship between genuine stress-induced stress granules and FUS cytoplasmic aggregates have not been addressed properly, and I'll try to do it in the first part of my talk. So to start with, we, as many others, overexpressed cytoplasmically mislocalized FUS either with point mutation in nuclear localization signal or with deleted NLS in different cell types, in neuroblastoma cells and in primary hippocampal neurons. And what I observed that in addition to cytoplasmic diffuse staining in some cells, FUS formed small dot-like granules which could further cluster together and form higher order assemblies, and in some cells larger, more compact aggregates. So these are just relative abundances shown here, and this process was clearly concentration dependent. On higher resolution images, stress-induced stress granules looked like amorphous structures, whereas FUS aggregates of the same size presented like higher order assemblies of small dot-like FUS granules, as we name them. And we confirm this pattern with FUS with different tags, such as TAP tag or flag tag, and TDP43 upon mislocalization didn't form that sort of structure. So FUS aggregates were reported to be positive for a variety of stress granule markers, just, for example, G3BP1 protein, and we also observed that in our studies. However, in a fraction of cells, FUS aggregates were completely negative for known stress granule markers, as just a couple of examples here. And furthermore, ribosomal protein S6, which is a component of stall pre-initiation complexes and which is abundant in stress granules, we could not detect it in FUS aggregates. Also, it is known that stress-induced stress granules, docked with with p-bodies. However, we did not observe such a coupling of p-bodies with FUS aggregates, shown here. So this data combined made us think that upon accumulation, the cytoplasm of higher than a certain concentration threshold, FUS gives rise, nucleates small FUS granules, which can form high order assemblies, which we called FUS aggregates. So we went on to characterize these types of structures. So firstly, we confirmed that RNA is an important structural component of FUS aggregates. Firstly, they are enriched in polyadenylate transcripts in RNA fish. And when we isolated granule enriched fraction, aggregate enriched fraction, it was sensitive to RNase A treatment, could be completely solubilized. And we also showed that stress granule-associated proteins, for example, tire, they're held within FUS aggregates weakly, probably on the surface via RNA interaction because RNase treatment on cover slips after mild methanol fixation could completely remove, for example, tire from FUS aggregates, and FUS can sequester tire into aggregate enriched fraction here, but it can be completely solubilized back by RNase treatment. Then we also show that FUS aggregates are dynamic and stable structures because they could be dissipated either by inhibition of transcription or translation by emitting actinomycin D. So they are dependent upon supply of both newly synthesized transcripts and transcripts running off of polysomes. Then using a set of truncation and chimeric proteins, we show that both prime-like domain and specificity of RNA binding are both required for FUS aggregation in the cytoplasm because when either of these criterion is not met, FUS does not aggregate and and is not recovered in insoluble fraction anymore. Sorry. So FUS aggregates composed of small particles, FUS granules. So what are these? Firstly, on high-resolution images, we observed they appear like uniformly sized particles of about 100 nanometers in diameter, and that actually corresponds to the size of RNA transport granules. So we thought that we can isolate them in the same way. So we used one of the established protocols for that, just schematically shown here, and we isolated a granule-enriched fraction, which was indeed detergent resistant and RNA sensitive, and the core protein of RNA transport granules in neuron FMRP also behaved similarly to FUS, although in immunofluorescence we didn't observe enrichment of FUS aggregates in FMRP. In contrast to FUS aggregates, which are so high order assemblies of FUS granules, FUS granules were not sensitive to emitting treatment to translational inhibition, but they were sensitive to actinomycin D treatment. So newly synthesized transcripts probably contribute to the assembling integrity. Then we wondered if there are proteins in neuronal RNA transport granules which can be recruited to FUS granules because they are structurally similar and behave in a similar way. So out of those proteins which we checked, we could find only one, which is RNA helicase DDX5, which was present in significant portion of FUS granules, and as expected, FUS aggregates which are formed out of FUS granules, they are also strongly positive for the DDX5, both in primary neurons and in neuroblastoma cells. Although DDX5 is not a component of stress-induced stress granules, as we also showed. So FUS aggregates, they are quite big and they recruit RNA in the cytoplasm, so one would expect that they would interfere with the integrity of other RNP bodies in the cytoplasm. So it was indeed the case because in those cells which contained FUS aggregates, if we stress them, they form fewer stress granules, and in such cells, P bodies were almost completely absent. And what we also observed that one of the core components of P bodies, DCP1 subunit alpha, was also recruited into FUS aggregates. So just quickly summarize here, upon reaching certain concentration threshold in the cytoplasm, FUS nucleates a novel type of RNA granule, FUS granule, which can cluster and form bigger structures, FUS aggregates. And by sequestration of RNAs and proteins, FUS can, these structures can disrupt the integrity of other cytoplasmic RNP structures, such as stress granules and P-bodies. So hopefully I convinced you that FUS aggregates are dynamic and stable structures, so they can constantly exchange RNA with the cytosol, and it is highly unlikely that they represent those unstable inclusions which we see in human FUSopathies. Sorry, I have a really bad hay fever. So we, as many others, are interested in the nature of second hit here. That is what makes these dynamic aggregates to transform, to be converted into irreversible and stable inclusions. Now, in our previous studies in cell models and in transgenic mouse model, we show that decrease in RNA binding capacity of FUS renders it more aggregate prone. And many of you are probably aware how difficult it is to achieve FUS aggregation in vivo models. And in these mice, we used an engineered protein with deleted C-terminus and the majority of RNA binding motifs, and these mice developed a robust proteinopathy in the nervous system and ALS-like phenotype. We wanted to go further and created another transgenic mouse in which RNA binding was less disrupted, because here we just deleted a portion of RM motif. And in these mice, encouragingly, we also observed proteinopathy, which was not although that pronounced as in the previous mouse line, but we observed multiple inclusions in the cortex and in the brainstem, and that kind of further supported our hypothesis. So we wanted to study that more systematically, so we created a set of construct to express proteins with gradually disrupted, FUS protein with gradually disrupted RNA binding. So as you see, RNA binding capacity decreases in this row, and what we've found is that aggregation propensity of the proteins nicely increased in this direction. So we show that using neuroblastoma cells and primary neurons, actually those variants with disrupted RNA binding, they aggregated even processes which we never observed for full-length protein or protein with only only minor disrupted RNA binding. These three constructs, these three variants were not able to sequester RNA, and they aggregated robustly, and they were not sensitive to RNase treatment, so they formed RNA-independent aggregate. Although neither of these variants, as we showed in SDDH, was of amyloid nature, which is consistent with non-amyloid nature of inclusions in FUS proteinopathies. Right, it was all great, but as you also were, the majority of FUS mutations, they don't affect RNA binding motifs of FUS, but very rarely it happens. So it is full-length protein which aggregates and forms inclusions. So we wanted to obtain more physiologically, or if you want, more pathologically relevant data. So we decided to model inhibition, decided to model RNA depletion by inhibition of transcription in cells which express fully RNA binding competent full-length protein. So to do that, we employed actinomycin D treatment, and in parallel, we monitored presence of polyadenylated transcripts in FUS aggregate. And what happened, so after 1 hour of actinomycin D treatment, the aggregates did not change grossly their appearance, but already after 2 hours of treatment, so the majority of the aggregates did not contain RNA anymore, as you see in RNA fish, and they appeared somewhat disintegrated, and after 3 hours of actinomycin D treatment, the majority of these aggregates was no longer RNA positive, and we observed also secondary aggregation of these RNA negative species. As a maximum, we treated our cells for 9 hours with actinomycin D, and this our time point, virtually all cells contained these large juxtanuclear aggregates, which resembled aggregates, well, normally one per cell. And biochemically, when we isolated aggregate enriched fraction, as we did previously, from cells which were treated with actinomycin D for 6 hours, we observed that it was not any longer sensitive to RNase treatment. So just to quickly summarize here, RNA binding regulates aggregation propensity of FUS, and slow deficiency of RNA can potentially lead to RNA-dependent aggregation of FUS in the cytoplasm. So based on these findings, we came up with a model, hypothetical model, which can potentially explain the chain of molecular events which can take place in the developed human FUSopathies. So under normal conditions, FUS is in the nucleus, and it is also a minor component of RNP particles, such as RNA transport granules and stress granules. But when it is shifted to the cytoplasm due to a mutation or post-translation modification or transporting deficiency, whatever is the reason, so it nucleates with granules or after reaching a certain concentration in the cytoplasm. And then, so this sequesters RNA, these structures formed, FUS aggregates, they grow, and stress facilitates their growth. We also have this data, which I didn't have time to show you. And even at this time point, the regulation of transcription, like global transcription, can take place because FUS is involved transcription itself. Also, it can be age-dependent decline in transcription. So many different factors. And at this stage, so it is transcription is attenuated, and the supply of new transcripts is blocked, and we can observe the transformation of dynamic unstable FUS aggregates into stable RNA-independent structures. So this is just conclusions once again, and for future directions, the PhD student in our lab, Hannah, is now trying to purify FUS granules and FUS aggregates, and including under conditions of transcriptional deficiency, and characterize their RNA and protein composition. And I want to acknowledge those who fund us and those who were involved in the study, and these are current members of the lab, and these are former members. And special thanks to my little son, who behaved and did not interfere too much with the study. And thank you for your attention. I'm happy to answer questions.